This method can help answer key questions in the HIV field, such as determining the genetic composition of HIV proviruses in different cell types, especially identifying those that are genetically intact and potentially replication competent. The main advantages of this technique is that it allows us to amplify near full-length HIV proviruses from single HIV-infected cells, and then sequence these by next-generation sequencing.
To begin, prepare all buffers as outlined in the text protocol. Obtain a cell pellet and add 100 microliters of lysis buffer per million cells. Mix by pipetting up and down. Lyse the cells by incubating in a thermocycler at 55 degrees Celsius for 1 hour, and then at 85 degrees Celsius for 15 minutes. To begin amplifying single HIV-1 DNA proviruses, mix the reagents for the first round PCR as listed in Table 1 of the text protocol. Add 38 microliters to 85 wells in a 96 well PCR plate, and label this plate as PCR1. After this, move the PCR1 plate to a clear area designated for the addition of genomic DNA. Using tris hydrochloride, serially dilute the genomic DNA from a ratio of 1:3 to a ratio of 1:81, making sure to prepare 45 microliters of each dilution. Add 2 microliters of diluted genomic DNA to each sample well and 2 microliters